Hey guys, welcome for chapter 13. This is going to be on meiosis, creating variation. So, if we look at living organisms, they are distinguished by their ability to reproduce their own kind. Organisms will inevitably resemble their parents, and that is because of heredity, the transmission of traits from one generation to the next. So, as a sexual reproducing species, that transmission of traits from one generation to the next would require gamete production, which is sex cells. Gametes are sex cells. And in order to create sex cells, you need meiosis. Now, as a result of meiosis, we also increase variation. Now, what is variation? It just shows that one offspring is going to be different to the everybody looks different, in other words. And that is also related to genetics, which is going to be the unit that follows this. Genetics is directly related to meiosis, and we'll show you how that is when we get into Punnett squares and stuff. So, first section, genes are the unit of heredity. They are a segment of DNA that codes for a trait. There is tens of thousands of different genes in the human genome, and each one will code for a specific trait or a specific protein. Cells will translate these genetic sentences, these genetic units of inheritance, in other words, to create features such as freckles, for instance, or hair color, any number of different things. And the sperm and ova will transmit these genes to the next generation. So each gene in an organism's DNA will have a specific locus. In other words, hair color, for instance, is at a specific spot on a specific chromosome, and it's there for every human in that species, okay, or for every organism in that species. Each gene will also be inherited. So you get 23 from each parent. So humans have 46 total chromosomes. However, it's essentially 23 pairs. You get 23 from your mom and 23 from your dad to create you, which is why you resemble them, but you don't look exactly like them. So those 23, again, they are pairs. So for instance, if you get like on the first chromosome, we'll say if that has hair color, that means you got a gene for hair color from mom, and you also got one from dad. Each chromosome has a single DNA molecule with hundreds to thousands of genes on them. There's really two strategies for reproduction. There's asexual reproduction, which could be done by mitosis. And in that case, because remember, mitosis creates identical cells, this bud, so this is a hydra, it's an organism that is reproducing asexually. This bud is a genetically identical copy to this parent cell. So there's no variation, at least theoretically. The only variation that could be in there would be from mutations. Okay. Versus in sexual reproduction, two parents would give rise to an offspring that is unique. It is varied. Offspring will vary genetically from their siblings as well as their parents, even though you get your DNA from them, essentially. So fertilization and meiosis are going to alternate in sexual life cycles. So a life cycle is just generation to generation, and we'll look at that. But in humans, each somatic cell, which a somatic cell is a body cell, which is essentially every cell in your body except sperm and egg. So sperm if you're a male, egg if you're a female. Every cell in your entire body is that. So again, a somatic cell is every cell in your body except for your sperm and egg. The only cells, again, that won't would be a sperm for a male and an egg for a human. So this would be a human karyotype, which is a mapping of our chromosomes. So you can see we get 2 copies of the first chromosome, one from mom, one from dad, okay, and so on for the rest. The only ones that are a little different are the sex chromosomes. So if you have 2 X's, you would be considered a female, an X and a Y would be a male. That would be you either an either or thing, obviously. So again, a karyotype is just a mapping of those chromosomes, visual representation. A huge term for this chapter is a homologous chromosome. And that is when, so again, like I showed you here, this would be homologous chromosomes. You've got one from mom and one from dad. So just another representation there. After replication, which would happen in interphase, they would now look like this. Well, this side and this side are exact copies of each other. Okay, so just be aware of that. And this is an exact copy of this side. They also would have the same gene. So going back to my hair color example, if hair color was right here on this chromosome, it would be right here on that chromosome. So they house the exact same genes. Now, might those genes be different? Sure. Like if dad has black hair and mom has blonde hair, you might have 2 different genes. Okay, but they have the same types of genes on them. Sometimes we call homologous chromosomes autosomes. Autosomes just means they're not sex chromosomes. Again, you just get one from mom, one from dad. So you have, that's why I said before you have 23 pairs of chromosomes. Now, as far as the sex chromosomes, we call those X and Y. So if you're a female, you have 2 X's. If you're a male, you have an X and a Y. This is why sometimes we say that males are the determining factor of if a child is a male or a female, because a female can only donate an X. A male can either donate an X or a Y, depending on the gamete that fertilizes the egg. And this is a joke. I'll let you read through those. This is a joke for the genes that are housed on the Y chromosome. So this would be for males, obviously. All right, diploid cell. A diploid cell has 2 sets of each of the chromosomes. In other words, this 2N means you have 2 sets. N is the haploid number, so 2 times the haploid number. So for humans, the haploid number is 23, so 2 times 23 is your 46. So a human has 46 chromosomes. You have 2 sets of 23 to get to there. All of your body cells, except for your gametes, your sperm or your egg, have this full 46 chromosomes. In a cell in which DNA synthesis has occurred, all the chromosomes would be duplicated and thus consist of 2 sister chromatids. And again, those sister chromatids, it's an exact copy on each side. So in meiosis, what would eventually happen is those homologous chromosomes would line up next to each other. So these 2 would be homologous, these 2 would be homologous, and these 2 would be homologous. How do I know that? The chromosomes are always going to be the same size. Homologous means they're of the same gene. So again, they would house those same genes. Unlike somatic cells, the gametes then would be considered haploid. Haploid is N equals 23. Again, N is that one set of chromosomes this time for 23 total. They only contain one set. For humans, again, that's 23. So during fertilization, the gamete, the sperm and the egg, would come together and reestablish that diploid zygote. Zygote is simply the first cell formed after fertilization, as it says here. So 23 from the sperm, 23 from the ovum or the egg, would reestablish that diploid organism of 46. So this would be the human life cycle. This would be the organism after fertilization, which it is important to point out that after fertilization, organisms develop by mitosis. So mitosis and development go hand in hand. So you would go from a single-celled zygote to an adult organism through mitosis, creating identical cells. So all of the cells in your body are identical unless a mutation happened, but theoretically they are at least identical. The only cells that might be different would be your sperm and egg. So the male and the female would create their sex cell through meiosis, and they would come together to reestablish again that fertilization, through fertilization, that new organism. So in animals, meiosis occurs during gamete formation. And we're not going to go through that part. All right. So now we're actually going to go through what meiosis actually looks like. Meiosis overall reduces the number of chromosome sets so you're going from diploid, the original cell is diploid, it's going to become a haploid cell in the end. You can create 4 haploid cells in the end. Many steps will resemble mitosis, so the steps of this will look very similar, and that I think should help you at least remember what's going on here. But there's going to be a couple key differences. So just an overview here, interphase still happens. So in interphase, DNA would get copied. So you can see now we have copied or duplicated chromosomes. In meiosis 1, we separate homologous chromosomes. So these would be a set of homologous chromosomes. Those were separated. And then in meiosis 2, we separate the sister chromatids. All right. So in meiosis 1, again, it is preceded by interphase. So the DNA would have been copied still. It's going to reduce the number of chromosomes from diploid to haploid. So in meiosis 1 is actually where we become haploid. We do not any longer have a double set. We only have a single set. And they separate homologous chromosomes. I'll show you what that looks like again in a little bit. Meiosis 2 then is where we produce 4 haploid daughter cells. And they would only, again, have a single set of chromosomes since being haploid. And that we separate sister chromatids in meiosis 2. So here's what that would look like. Interphase looks exactly like it did in meiosis. Why? Because this is essentially a diploid cell, which is what we normally would have. Now, as far as like the behind the scenes stuff that is happening in prophase, metaphase, anaphase, it's the same pretty much. So the nucleus would disappear. The centrosomes are moving to opposite poles. The DNA is going from chromatin into chromosomes. All that stuff is still happening in these phases, okay? The difference is you'll see this is called prophase 1, metaphase 1, anaphase 1. The difference is we now have homologous chromosomes, okay? So they're going to line up next to each other. Now, prophase 1 is the most important phase in all of meiosis. It's where really the variation comes into play. What you're seeing here, and this labeled as a chiasmata, is crossing over happening. I'm going to go through a little bit more on that later. But essentially what that is it's trading genetic material. And you can see that. See, it's still crossing over here. But when they pull apart, you'll see this chromosome is mostly red, but it has a piece of blue, and vice versa for this one. So they have swapped genetic information. Essentially, remember, say if this blue was dad's chromosome that you had, now it has a piece of mom's chromosome. So we've made a brand new chromosome, essentially. So that's very important that it happens starts to happen at least in prophase 1. They come together, those homologous chromosomes. In metaphase 1, those homologous chromosomes are going to line up along the metaphase plate. So M still stands for middle. And when they pull apart, A still stands for apart or away. They will be pulled apart. But what are we pulling apart in this case? We're not pulling apart sister chromatids. We are pulling apart homologous chromosomes. So make sure you're aware of that. Now, as we move into meiosis 2, this will look a lot like mitosis. It's just mitosis times 2 almost, right? That's what it looks like. But what we'll notice is in the end, these 4 cells, 1, 2, 3, 4, they all have a unique set of DNA. And if you look at them, they are different. Okay, so the 4 cells that we're creating are different at the end. But again, what did we pull apart here in anaphase 2? Meiosis 2 separates sister chromatids. So meiosis and mitosis can be distinguished by 3 really key events. And we'll go through those now. The first is synapsis and crossing over. So if we look at these chromosomes, as I said, crossing over is kind of an exchange. So if the green is dad's and the yellow is mom's chromosome, we essentially made 2 brand new, never seen before chromosomes right here by crossing over. So they have essentially traded spots. Okay. The tetrads on the metaphase plate at metaphase 1 of meiosis paired homologous chromosomes, and they are positioned. It is an equal chance that blue is on the right side or blue is on the left side. So essentially what we're saying there is it's a 50-50 shot which side of the cell gets the dad's chromosome or the mom's. And we'll go into more on that in a minute, but it is homologous chromosomes that are on this metaphase plate during metaphase 1. It's not just sister chromatids anymore. It's homologous chromosomes. And the separation of homologous chromosomes would happen in anaphase 1, where sister chromatids separate in anaphase 2. It's important to be aware of that, and it really creates some key distinguishings. So if we look here, what we would call this is a double division. Okay, so we created 4 haploid cells, a reduction division as well, versus, again, mitosis, we created identical cells. So the parent, if we look at the parent cell here, the parent cell here is exactly the same as these. That's mitosis, key sign, identical, versus the parent cell is different from here. And these 4 cells, if you look, they are also different. If you want to review those phases of meiosis, I encourage you to watch this video done by Mr. Anderson. All right, so let's look at how meiosis creates genetic variation. It's important to point out that the original source of all genetic diversity is mutation, but mutation is really random. It's not really all that organized. It's just kind of random chance. And doesn't create nearly as much genetic variation as meiosis would. Meiosis and fertilization produce further variation by producing offspring that is unique to anything that's been seen before, at least for the most part. In species that produce sexually, the behavior of the chromosomes is really what's creating this variation. We'll go into that right now. So the 3 things that are really, really important, and these are the 3 mechanisms that are going to create genetic diversity. The first one is independent assortment of chromosomes. And this would be how the homologous chromosomes line up or orient along the metaphase plate. Then we also have crossing over, and then we have random fertilization. So I'll go through each one individually. So again, I mentioned this before, but it's 50-50. Possibility 1 is that they would line up blue on the left, red on the white, and then if you look at the bottom one, it's the red and the blue flipped here. Okay, so it's 50-50 chance. Now, that might not seem like that big of a deal, right? But in humans, we have 23 sets, so it's 50-50 for each of those 23 sets. Essentially, and this is kind of crazy to think about, but this creates 8 million different combinations. How did I figure that out? Well, essentially, the formula is 2 to the nth power, where n is the haploid number of the organism. Let me write that down for you on here. So this is an important concept, and you'll see it on the test. So what the formula we have here, again, is 2 to the nth power, and n equals the haploid number. of that organism. So for humans, that equals 23. Okay, so how did I get that 8 million? Well, it's 2 to the 23rd power. It's not 8 million exactly, but it's more than 8 million combinations of chromosomes. So just by this alone, that 50-50 orientation, again, that doesn't seem like maybe that big of a deal, we created over 8 million different combinations. So pretty crazy to think about, and that's just the first one. Now we have crossing over, where we're creating completely new chromosomes by essentially combining mom and dad chromosomes. So again, the red from mom, blue from dad. But if you look here after they've separated, now this blue one has pieces of the red. So just creating further genetic variation, because they're essentially have a chromosome derived from two different parents now. And the last one is called random fertilization. So remember from the independent assortment one that we just went through, that 50-50 one, we created over 8 million different possibilities in a single gamete. Now we have random fertilization, where we can produce a zygote with about 64 trillion different combinations. And that's before even considering crossing over. This is just from random fertilization and independent assortment. How do we get that? It's 8 million times 8 million. Because remember, we have an egg that had 8 million different possibilities. We have a sperm that has 8 million different possibilities, equals about 64 trillion different combinations. And that's before even accounting for crossing over. So in other words, it's darn near impossible for two organisms that are humans to look exactly alike, because there's over 64 trillion different combinations. So huge numbers. So just to review, independent assortment of homologous chromosomes during meiosis 1 is the first thing that can create variation, and that created 8 million different possibilities, 2 to the haploid number for humans. That would be 2 to the 23rd for over 8 million different possibilities. Crossing over exchanges material in the homologous chromosomes from mom and dad, so essentially creating brand new chromosomes that have never been seen before. And then we have the random fertilization, where any sperm could fertilize any egg, and that creates that over 64 trillion different possibilities. So crazy amounts of variation from essentially sexual reproduction and meiosis. Genetic variation is the raw material for evolution, because there has to be variation. We'll talk about more on this later in the year, but there has to be variation in order for natural selection to happen. Adaptation absolutely depends on it. And mutation, while it is the original source, sexual reproduction has definitely added to the genetic variation on our planet. And with that, I have finished meiosis. We'll review this week. Have a good one.